This pre-lab talk consists of two videos. In the first, I will give an overview of colon cancer, and in the second video, I will consider the individual slides and describe some of the significant structures that you should examine. The learning objectives for this laboratory are that by its conclusion, you should be familiar with the normal structure of the colon, be able to identify an adenomatous polyp as well as invasive adenocarcinoma, and finally understand how to stage colon cancer. In the present video, I will cover some of the general features of colorectal cancer as shown in this slide. First of all, it is the second leading cause of cancer death in the general population of the United States. Now, previously I had told you that breast cancer is the second leading cause of cancer mortality in women, and that prostate cancer is the second leading cause of cancer death in men. However, both of these are gender specific. If you take the population as a whole, then colorectal cancer is the second leading cause of death. Of course, lung cancer remains the leading cause of death in whichever population you look at. The major risk factor for colon cancer is basically aging. Indeed, the incidence of colon cancer begins to rise at about the age of 45 and increases dramatically up to the age of about 80, where it begins to level off. This is why screening by colonoscopy is recommended for individuals over 50 years old. Now, the vast majority of colorectal cancers are adenocarcinomas that arise from the stem cells in the epithelium lining the crypts of Lieberkühn. More on this later. Now, this slide shows the gross anatomy of the colon. Please note that there are three major sections of the large intestines: the ascending, the transverse, and the descending colon. Now, the function of the colon is to remove the water and salt from the chyme after most of the nutrients have been absorbed in the small intestines. As you might imagine, water and salt were valuable commodities for our ancestors, so being able to retain these substances makes evolutionary sense. This figure shows the structure of both the small and the large intestines, the small on the top and the large on the bottom. You should note that their basic structures are quite similar. Indeed, the wall of the GI tract follows the same basic pattern throughout its entire length. Basically, it consists of four layers. From the outside to the inside, these are the serosa, the muscularis externa, the submucosa, and finally the mucosa. What I would like to do now is describe each of these four layers in greater detail, since they will be important when you go to stage colon cancers. The outermost layer is the serosa. It is considered to be a type of serous membrane. This is in contrast to the mucous membrane that lines the inside of the GI tract. Now, the difference between these two types of membranes is that a serous membrane lines cavities in the body that do not have openings to the outside. This includes the pleural, the cardiac, and in this case, the peritoneal cavity. Mucous membranes, on the other hand, line cavities that have openings to the outside of the body, such as the respiratory and the GI tracts. Another difference is that mucous membranes often contain mucus-producing goblet cells, while serous membranes do not. Returning now to the serosa of the colon, it consists of two layers: an outer epithelium of simple squamous cells and an underlying layer of loose connective tissue. And generally, this includes a large proportion of adipose tissue. The next layer is a muscularis externa. This consists of two layers of smooth muscle oriented at right angles to each other, circular and longitudinal. The coordinated contraction of these muscles is responsible for the peristaltic movement of the GI tract that propels the chyme along its length. In places between these two layers of smooth muscle are ganglions, which are collections of neuronal cell bodies. Inside the muscularis externa is the submucosa. This is composed of loose connective tissue that is lighter staining than the smooth muscles of the muscularis externa. You will also find blood vessels within this layer. The final innermost layer is the mucosa. As I said before, it is a type of mucous membrane. It has openings to the outside of the body and contains mucous goblet cells. Now, as you can see in this diagram, the mucosas of the small and the large intestines have a slightly different structure in keeping with their different functions. Note that the mucosa of the small intestines has small finger-like projections called villi that increase the surface area to maximize the absorption of nutrients. In contrast, the surface of the colon is flat because all it does is absorb water and salts, which does not require a large surface area. Now, the mucosa consists of three layers: the muscularis mucosae, the lamina propria, and the lining epithelium. The outer boundary of the mucosa is defined by a thin layer of smooth muscle termed the muscularis mucosae. In some areas, it is interrupted by lymphoid nodules, which are collections of lymphocytes. These are normal structures that function to guard the mucous membrane from infection and are transient in nature. They come and go. The lamina propria is a layer of loose connective tissue that is found between the muscularis mucosae and the epithelial lining. It contains blood vessels, smooth muscle cells, fibroblasts, and cells of the immune system. The final component of the mucosa is the lining epithelium that is composed of simple columnar cells and contains numerous mucous goblet cells, which I will describe further in a minute. Please note that while the surface of the colon is flat, it does contain numerous gland-like structures called the crypts of Lieberkühn. This is a diagram of one of the crypts of Lieberkühn. It turns out that the stem cells that give rise to the epithelium lining the colon reside at the very base of the crypts, shown here in yellow. Remember that when a stem cell divides, one of the daughter cells renews itself, while the other goes on to differentiate. This latter daughter cell migrates up the side of the crypt, and as it does so, it continues to divide and differentiates into both absorptive cells and goblet cells. Now, the goblet cells got their name because they simply resemble goblets. They produce mucin that protects the surface of the colon. Now, this mucin is washed out of the cells during processing for histology, so that they appear to be as empty spaces. Most of the cell division takes place near the base of the crypts. These cells then migrate across the basement membrane and reach the surface. They are eventually sloughed off the surface and replaced by younger cells. Now, the relevance of all of this is that the stem cells and their immediate progeny at the base of the crypts are believed to give rise to the vast majority of colorectal cancers. Another important aspect of colon cancer is the role of polyps. Now, a polyp is an abnormal growth that protrudes from the surface of a mucous membrane, which includes the GI tract, and it may or may not be attached by a stalk. There are two types of polyps. First, there is a simple hyperplastic polyp, in which all the epithelial cells are morphologically normal. That is, they are similar to the regions labeled normal in the right-hand figure. Note the large number of goblet cells. These are the most common types of polyps and do not indicate an increased risk of cancer. The second type is an adenomatous polyp, and this is of greater concern. Such polyps contain dysplastic epithelium, that is, the cells are not quite normal. An example of dysplasia is indicated in the right-hand figure labeled adenomatous. When you compare this region to the normal regions, you will note that it is darker staining, the nuclei are more elongated, the nuclei are not just located at the base of the cells, and there are fewer goblet cells. If the polyp contains regions such as this, then it is defined as an adenomatous polyp. Remember, the term adenoma simply means a benign growth of a secretory epithelium, so this is considered to be a benign condition, but it can lead to cancer. Indeed, most colon cancers arise from adenomatous polyps, and the larger the polyp, the greater the risk. The occurrence of these polyps increase with age. Also, they easily bleed, and this is why stool samples are often checked for occult blood. This slide shows the structures of polyps, ranging from normal hyperplasia to invasive adenocarcinoma. The structure labeled 1 is simple hyperplasia and does not indicate an increased risk of cancer. Structures 2 and 3 represent adenomatous polyps, in which the epithelial cells show some signs of dysplasia, but this condition is still considered to be benign. Structure 4 is close to the borderline between benign and malignant. It contains a region of severe dysplasia shown in white here, but this is confined to the mucosa. It has not passed the muscularis mucosae. You might also think of this as being carcinoma in situ. In structure 5, the region of dysplasia has invaded past the mucosa and entered the submucosa. Passing this threshold means that it is now considered to be malignant. And finally, in structure 6, the polyp is gone, but the adenocarcinoma has passed through the wall of the colon and into the peritoneal cavity. Now, one important consideration in this progression is that if this condition can be caught early on, then it can be corrected. Suppose that the adenocarcinoma has not spread too far, such as in structures 1 through 5, then it can be removed by colonoscopy. The figure on the left shows what a polyp looks like as seen by colonoscopy. The polyp can then be removed using a snare-like device as shown on the right, after which it can be sent to pathology for further analysis. If no adenocarcinoma is present in the stalk, then no further treatment is necessary. Now, this approach for removal of precancerous polyps has reduced the incidence of colorectal cancer in the United States since it came into wide use in the late 1990s. In this laboratory, you will be asked to stage some of the tumors. Now, in colon cancer, staging is considered to be far more important than grading for determining the prognosis and treatment. Remember, an important aspect of staging is the extent or spread of the tumor cells. Staging of colon cancer depends on whether the tumor cells have passed certain landmarks in the wall of the colon, and this must be evaluated under the microscope. This figure shows part of the TNM system for staging cancers, and it is the T part of this system, the spread of the primary tumor, that we will be concerned with here. In T1, the adenocarcinoma has not passed the landmark of the muscularis mucosae. In T2, it has invaded into, but not past, the muscularis externa. In T3, it has passed into the serosa, and finally in T4, it has gone past the wall of the colon and entered the peritoneal cavity and perhaps adjacent organs. Please keep these landmarks in mind as you examine the slides today.